Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is October 17th, 2024, and we're talking with Eric Fortune, who is an associate professor of biological sciences at New Jersey Institute of Technology. Eric studies the mechanism of evolution and animal behavior, which sounds a little vague, and that's because it's hard to summarize his work in a kind of a bumper sticker. He studies natural animal behavior in a natural environment, usually in some kind of exotic location, and animals that live there, and he does his experiments there. And he combines measures of behavior with neurophysiological studies of neuronal activity, often done in the wild, which is kind of unusual. And he was here before in September 2013. At that time, he talked about songbird duet singing in South America, somewhere, Ecuador?
In Ecuador,
In Ecuador. Right. And that podcast is episode 102 for anybody who wants to look it up. I listened to it today, and it's pretty good, although the sound quality is not as good as we would be satisfied with now. Eric still works on duet singing, and we could visit that topic today if we get around to it. But I think we're going to mostly talk about active sensation and weakly electric fish, which is another one of his ongoing research interests. Hi, Eric, and welcome back.
Thanks so much for having me.
Also with us is Francesco Savelli, our local computational biologist, expert on animal navigation in space and time in the brain. Hi, Francesco.
Hello.
And Todd Troyer, a birdsong expert and longtime podcast colleague, who was actually here for Eric's first podcast.
Yeah, I'm happy to return.
Todd. And me, I'm your host, Charlie Wilson. So, Eric, our listeners know something about weakly electric fish. Harold Zacon was here 3 years ago and talked to us about evolution of the electric organ, which was really cool. And but let's still, if we could get you to give us a quick review of the electric sense in these fish and what's cool, what the fish uses it for, what's cool about it, what attracts it usually.
Yeah. Well, Harold Zacon is one of my academic heroes. So he's just done amazing work and has been a real inspiration for me, although the work I do is quite different from his. And our interest probably started at the same place, which is that weakly electric fish seem like an animal where you can actually solve the problems, answer the questions that we're interested in. So I started my graduate career with songbirds, and singing is a really complicated behavior. It's complicated to perceive. Sound is complex. But in weakly electric fish, these animals, the species that I study, they produce an electric field, and the electric field uses the same currency as the nervous system. So it's there's no transition to waveforms of sound, or you don't even need movement for this for electrical behaviors to occur. It's electricity, the same thing that the nervous system is using for its own control. And then on top of that, the species that I look at are called wave-type weakly electric fish. And so they produce a sinusoidal, or almost perfectly, it's not a perfect sine wave, a sinusoidal electric signal. And because of that, the signal itself is super simple to study. We can use frequency analyses, this whole suite of tools, to understand how the behaviors work. And that's really what attracted me when I first started working with them, that they had these seemingly complex behaviors all mediated by this hyper-simplified system that was as close to the nervous system as you could get. And when I was a postdoc, the other reason why I was initially attracted to them was for a very simple technical issue, a neuroscience-y technical issue, which is that back then it was difficult to do intracellular recordings from awake behaving animals. And my postdoc advisor had just devised a technique for doing just that. So you could make intracellular recordings from the neurons that were controlling the behavior while the animal was performing the behavior. And back in the 1990s, that was an enormous technical feat and a new way of analyzing neural control of behavior. Now, of course, people do this in rats and all sorts of other awake behaving animals, so that technological advantage isn't there. But, more recently, we've been sort of using, instead of sort of using high technical advantages, we've been applying a scientific perspective on analysis to these animals, which has been pretty productive.
So as the fish is swimming along, it's making this sine wave, and then it senses its own sine wave.
Yeah, so how does the electric, that was a lot of drifting before getting, so how does this electric field work, right? So the animals have an electric organ. The electric organ is in the side of the animal and in its tail. And for most species, it's a modified muscle cell. And when we say a modified muscle cell, it means that the actin and myosin no longer is inside the muscle cell, so it doesn't contract. And all of the sodium and potassium channels have been moved to the ends of the cell. So if you line them up when they fire, they act like a little battery in series. So all of these cells are on the side of the animal. The skin is super high resistance, and so when all of the cells of the electric organ fire, they produce a potential difference between the inside and the outside of the fish, because the skin has high resistance. And the pathway for the current to come back to the other side of the electric organ is through pits in the skin that are filled with electroreceptors. And so the electroreceptors are the low resistance pathway, and so they detect the voltage difference between the inside and outside of the fish, and they provide the return pathway for the current. And so the electric field in the water extends, roughly about, depending on the conductivity of the water. So the conductivity of the water is related to how much rain there's been. If there's been a lot of rain, the conductivity will be low. And if there hasn't been much rain and there's more salt in the water, the conductivity will be high. And so in low conductivity water, the electric field can reach out for more than a meter around the fish. Higher conductivity, it will be smaller. And so if there's fish that are within a meter of each other, their two electric fields will mix together and they can detect it with their electroreceptors. So both fish inevitably will detect each other.
So they live in tidal water so that the salinity of it changes all the time?
Well, it varies a lot depending on the species and where they happen to live. There's more than 300 species of weakly electric fish in the Amazon basin, and there's another hundreds of species that have evolved independently in Africa, and I really focus only on the South American species, so we'll just stick with them. But they're found everywhere. So there are species that are in the wide open center of the Amazon River proper, but where I find them mostly are in black water, slow-moving streams and ponds, and we'll find them in habitats from the sides of small streams, in floating rafts of plants, or in root systems of old trees and things like that. And even in mud flats that are not tidal isn't quite the right word to use, but they flood and don't flood depending on rain. So I had an Ecuadorian student who went and measured the conductivity in and around the Yasuni National Park, which is a preserve in the center of the Amazon in Ecuador. And she found that the waters there varied between like 5 microsiemens, which is a measure of how conductive the water is, to maybe 50 or 60 microsiemens. And that is extraordinary, right? So when I had a problem with my reverse osmosis, the super expensive water that we get out of the building. And when we tested it at Johns Hopkins, it was coming out of the wall at 30 microsiemens, and so double the conductivity of what was typical for the fish in the field. But the conductivity, if there's not been any rain, the fish can end up being stuck in mud flats, and then the conductivity can be on the order of thousands of microsiemens. But most of the time, I would say for most of these animals, it's pretty low. It's, below 50 microsiemens or something like that. The purest water you could buy. If, you buy a bucket of pure water to put into your car engine, the distilled water, the microsiemens of that is going to be roughly what it naturally occurs in the Amazon.
So why are there so many species? Is it the habitat stuff, or is it the fact they use this for communication?
Well, I mean, I can speculate on that, but I don't know the answer. But I can give a couple of. First off, I mean, they live in the Amazon, and so what do you know about the Amazon? Is it's the most speciose place on Earth, and so the conditions of the Amazon basin give rise to microhabitats and reproductive isolation, which gives rise to speciation events, is my understanding of how all of that works. But at least part of it, I think, has to do with the electric signaling, the social electric signaling that they do. So just like songbirds have a lot, if you look at all of the species of birds in the world, songbirds are a lot of species. And I think that's in part because of the reproductive isolation that this social signal that the learned song provides them. I think with weakly electric fish, it's very easy for them to change the electric signal. A few small changes in the speed of a conductance of a sodium channel in the electric organ will have a behavioral impact on the frequency of that signal that's occurring in the field that they can detect. So on the production side, they can easily change their signal evolutionarily, simple changes in the kinetics of a channel, of channels. And on the perception side, the receptors themselves are broad spectrum and also have a lot of evolutionary variation. And so I think they also detect those differences quite easily.
So maybe there's a connection. Is that the connection, social communication with the birdsong stuff?
Oh, yeah, absolutely. I mean, it's interesting for my own personal scientific development. I don't know why it took me that long to figure it out, but I've been studying a particular behavior a social behavior in weakly electric fish, the jamming avoidance response. And that's a behavior when fish get near each other and their electric fields are near the same frequency. The interference patterns that arise from them being near the same frequency impairs both of their ability to electrolocate. So in some species, the fish will change their frequencies to avoid those patterns of negative interference. So the fish with the higher frequency will raise its frequency. Sometimes the fish with the lower frequency will lower its frequency, and then they'll be far enough apart so that the interference isn't. So that's fundamentally a social behavior. But it was only when I started working with these duetting songbirds that I realized just how important this exchange was, that fundamentally I'm studying a social behavior in which two actors are actively participating to negotiate the outcome of their pairwise behavior.
And they use this.
That made any sense.
That made sense. They use this to identify conspecifics. Is that part of the electric?
Well, I mean, I don't know how well the individuals are identifying each other based on their electric signals. Like, I just don't know how readily, in songbirds, it's really clear that birds can recognize individuals, right? You can do a neighbor-stranger discrimination experiment where the two birds are right next to each other. You can record their songs and then play back one of the two birds' songs, and they'll both ignore it. But then if you modify the song so it's a slightly different, then at least one of them is going to detect that it's a different bird and aggressively defend their territory. And I'm not aware of that kind of experiment being done explicitly in weakly electric fish. And part of it is because they just aren't as territorial in that way. And when they do fight over territory, they get physical. So they'll come in and they, have these beautiful battles where they go face to face, or some species can open their mouths really wide. They also have social signals that they generate that are aggressive. So they have these things that we call chirps, where they modulate the electric field briefly. And sometimes chirps can be aggressive, and sometimes they can indicate that you're the loser. And so people have shown through, and my lab a little bit as well, has shown that, when they're in the field, you can see animals interacting with each other. And then when one flees, they produce sort of a leave me alone, or I lost, or some other kind of signal that indicates that one of these types of chirps that says, hey, leave me alone, or something. I don't know what it means, but they produce it when they're losing.
But they're crying uncle.
Yes.
In your experiment, they're using the electric field just to locate themselves in space. Is that?
Yeah. So the evolution of weakly electric fields, almost, I think, was generated in terms of social signaling. So you can imagine, there are fish that have electroreceptors, but they're not able to produce their own electric fields. And the male is trying to attract a female, and his skin, for whatever reason, is leakier than some other skin, electrically leakier, so that when he bends his body, there's more electric field in the water that is then more easily detected by a female. And so he becomes a more salient signal. And you can imagine how that could lead to sexual selection, leading to the evolution of an organ that specifically drives the electroreceptors of females, increasing their salience and therefore leading to increased matings. That's a just so story that's made up, but I think it seems pretty reasonable. And then secondarily, I think the generation of weak electric fields was then used for locating objects in the environment. So the original array of electric electroreceptors found on the surface of the animal, like on sharks or other aquatic animals, is also used for detecting electric fields that just naturally occur in the water. Like a shark detects you emitting electricity as you flex your muscles in the water, and so it can detect those electric fields and come grab you. And weakly electric fish or other fish also do the same thing. And so the electric field that they ended up producing will interact with objects in the environment, so they can use that information to detect and characterize objects in their environment.
And they use it for prey.
Yeah, so there's some really elegant experiments by Malcolm McIver from the 1990s where he put electric fish, these weakly electric fish, in complete darkness, illuminated it with infrared light so that he could see them, but they couldn't see, and then dropped Daphne, a small prey item, and then you could see the animal go through and capture the prey using its electric sense almost exclusively. And so the fish will also use their electric sense to sort of navigate through their environments at night. They're nocturnal animals. And the experiments that I do take advantage of the fact that the animals hide in refuges during the day especially, but also sometimes at night. So the Amazon has a lot of predators. And so these fish, if you have them in the lab and you give them a tube or some other place to hide in, they will aggressively hide in these spaces, really go for it. They'll fight over them if there's more than one individual to stay out of the open areas of the tank. So much so that there's a product, so people buy these fish, the enthusiasts, pet owners will buy them. And one of the problems with these fish is that they hide all the time. And so there's a transparent PVC tube called You Can See It or something like that. And the fish will hide in the transparent tube because electrically it feels safe to them and physically it's safe for them. And so then you can see your pet, which is slightly better than if you can't see the pet, I guess.
So in your experiments, you try to fool them by moving the little tube and keep them.
I mean, anyone who's studied these animals has used the tube as a way of catching the fish. So if we're going to do a neuroscience experiment on an animal, we're going to have to take the animal to give it anesthesia or do whatever manipulation we're going to do. So you have to catch it from the tank. And of course you can try and take a net and try and catch the animal, but the animals are always in the tube. So you can then pick up the tube, grab the tube, move it through the water and place it in front of your net, and then lift the whole thing, and the fish will fall into the net, and then you capture it. So you take advantage of its hiding. And so I certainly wasn't the first person to figure out that the fish moved in these tubes. And my postdoc advisor, Gary Rose, he had set up a tube on an actuator, and he was training the fish. He had a second device where he could move a little object that the fish could detect with its electric field, and then a moment later he would translate the tube forward or backwards. And if you predict the movement with this little object, the fish would learn how to do it, so that you would give it the signal and it would move forward whether or not you moved the refuge forward. And so when I got my first faculty job, I set one of those up, thinking that I would do some experiments where I looked at the movement of the fish back and forth. But all of that was transformed by my meeting this electrical engineer, mechanical engineer, Noah Cowan. And we met because my daughter got locked out of my house. My daughter at that time was like 9-10 years old, and she had babysat for someone across the street. They had since moved away, but she got locked out of her house, and so she didn't know what to do. So she went across the street to the same house, knowing that the people had moved away. But it turned out Noah Cowan was living there, and they got to talking as she was sitting there being upset about being locked out of the house. And she said, "Well, my dad's a professor at the same university you are." And so we decided to meet each other. And so first thing he did was come over to my lab, and I showed him this apparatus with the fish moving back and forth in the tube. And I didn't get the response I was expecting, because he was ecstatic. He wasn't like, "What are you doing? This is idiotic." He was like, "This is my dream come true." He's like, "I've been waiting to do a whole animal body Bodhi plot my whole life." It's like, "What is a Bodhi plot?" was the first thing I said. But he wanted to perform a set of engineering analyses for understanding how animals control their behavior. And this was exactly the right behavior for his analysis. So at that time, we agreed to have that we wanted to work together. And so we put together a little atelier, a little teaching session for each other. So he made a notebook for me about control theory and engineering approaches that he used, and I put together a little basic neuroscience and behavioral neuroscience packet, and we both taught each other for a couple of months, our perspectives, and then we went on to do a set of experiments where we took the fish, and it was a really simple experiment where you have the tube, the fish is hiding in the tube, and we had a video camera that measured the position of the tube and the position of the fish, and we moved it back and forth at different frequencies, from low, really slow moving, 0.1 hertz or so, up to really fast, which for us was like 2 hertz. And we he did the analysis, and I sort of understood what was going on. And so we published our first paper together in 2007.
So what you're measuring is the amplitude of the fish movement.
Yeah.
Right?
So we move the refuge back and forward maybe 2 or 3 centimeters at a particular rate. And so then you can measure how far and how well the fish tracks. So if you move it really slowly, the fish is going to move perfectly along with the amplitude and with no phase lags, the movement of the refuge. But if you move it really fast, the fish can't keep up. And so the distance that it'll cover, the velocities that it generates, won't match the movement of the refuge. And on top of that, they'll have phase lags. And so the phase lag, if you move it forward, it'll take it some milliseconds to catch up, and back the other direction. And so you can see the phase lag. And so those are the 2 parts of this thing called a Bodhi plot, which is the gain. What's the relation of the movement, the amplitude of the movement of the shuttle to the movement of the animal? and then the phase part, which is how much time lag or phase lag it has as you change the frequencies. And what I didn't know at the time was that engineers can look at the patterns of those and understand a lot about the underlying control. And so in our case, the animal had what we call a low pass. They did really well at low frequencies, but not so well at higher frequencies. And the phase shift over that range of frequencies that we tried were a little less than 90 degrees. So we could say that it's a first order system, and some other details about how the animal performed. And so then
So that usually means there's a single characteristic time constant for the animal's behavior. So what limits the fish? Why can't the fish keep up?
So that's been the focus of our work then together for the past 20 years almost now. So there's a lot of parameters. There can be problems with sensing. It could be that the animal can only detect certain levels of changes, just like if you have a cruise control on your car, they don't maintain the rate of your car exactly at the right level. They have a little bit of variance around the set point. So maybe the animals can't detect all those frequencies equally well. Another possibility is that the animal has it uses its ribbon fin to swim back and forth, and it could be that the ribbon fin simply can't move fast enough, or they can't change direction fast enough. It also could be that there's drag on the animal, so they can't overcome the drag forces on the animal. And so we did things sort of in a stepwise way. We've been looking at both the sensory side and at the motor side separately, and we had sort of we also refined the control model that we developed over the years. So I'm really embarrassed talking about all of this because I know how much I'm saying wrong, and Noah will be shaking his finger at me because I bring the wet stuff to this equation, and he brings the math and.
But from a physiology perspective, the steps are basically: I send out my electric signal, I get an answer back.
Yeah, I
have to interpret that, turn it into a velocity,
motor signal.
Yeah, and then I have to decide how much to move.
Yeah.
And then what is the time lag for the sensory side of it? Is it fast?
Well, I mean, one of the interesting things about biologic systems that are different from engineered systems is the just enormous delays that nervous systems have. It's just shocking how many tens of milliseconds are built into each transformation inside of the brain out to the motor system. And that we can manage to do any of the amazing behaviors we do is just shocking to me. It really just is a testament to the solutions of evolutionary processes. So for the electric sense, I mean, the neurons that we record from, which are just 2 or 3 synapses away from the electroreceptors, the delays are 50, 60 milliseconds by that part. And the delays from a sensory input to a motor output are much longer, on the order of 100 to hundreds of milliseconds. And so these animals are performing at rates just like we are, where we can move at a few hertz. What's the fastest someone can tap? Maybe 4 or 5 hertz if they really are going forward, or if they're a drummer, they can go faster because they're using the resonance of the system. But we're limited to a few hertz, right? So if you're moving at 1 hertz, it's 1 second per cycle. And if you're moving at 10 hertz, it's 100 milliseconds per cycle. So that gives you an idea that there's a fundamental limitation of sensory motor processing that's going to be somewhere in that range.
So you mentioned that they're not very adaptive to, like, I don't know how much or what the time frame is adjusting to a sinusoidal movement. Because of the delays and being efficient, one of the ways to overcome that isn't to anticipate, right?
Yeah.
To do it before it happens. And then if you're slow, it doesn't matter if you're ahead of the game. So do you think they, I don't know, is it they do learn something and change their phase over long times, or maybe it's just so weird that they don't do it?
The movements are predictable, right?
Yeah, so I mean, we talked about that, or I mentioned it a little bit in the talk today, which is that one of the experiments that we did a few experiments that sort of explore this issue, which is we give them a predictable movement. So you can do a sinusoidal movement back and forth. And if they were really smart, so if you were in this situation, you probably would just stop moving and stand still. But the fish don't do that. They keep moving back and forth, even though there's no net translation of the refuge. But if you give them a unpredictable stimulus, so you can play something that's noisy, or sum of signs is a really nice unpredictable stimulus for them, they will be more aggressive about following the position and the velocity of the refuge than if you give them a predictable one. So they clearly are figuring out that there's something predictable here and reducing the amount of effort that they're putting into tracking the refuge. But they don't stop tracking the refuge, which is good for our experiments because they potentially could, but they don't. And this transition from less effort for a predictable stimulus to more effort for an unpredictable stimulus, we were really interested in how long it takes them to make that adjustment and how they learn or detect it. And it's just remarkably fast. It was far faster than we ever anticipated it. I mean, it's nearly instantaneous. Once it's unpredictable, the moment it's not predictable, they are believing it's predictable, and then as soon as it's predictable, they've already altered their behavior.
So I think in the talk you mentioned something about the approximational linearity, but now what you're saying essentially means it's not linear. You know, you can break down the unpredictable stimulus based on frequency component and predict the response based on the...
It's not completely linear.
It's not completely linear, which is not too surprising about, a system. But so I was just kind of thinking about like the open, what that impact could be with the open loop versus closed loop experiments that you were doing, because I also happen to be a collaborator of NOAA.
Oh, that's right. Yeah.
Like, so we actually did experiments. They're kind of very similar in a way to the ones you did, but with place cells. And so, I could talk, about what the closed loop gave us. As a matter of fact, there was just, a paper published this year. But what did bring to your system, this kind of analysis? What did you gain in terms of understanding the system about? I was thinking maybe you could talk a little bit about this aspect, because it's very intriguing.
Well, you brought up a lot of different things there. So, on one hand, you can have a fish in a refuge and move it back and forth. And it's a linear system as long as the fish, you move the shuttle at one frequency and you get the same frequency movement out of the fish. That's a definition. And then, if we do different frequencies and then we play those frequencies at the same time, we should get the same exact response. That's the superposition component of linearity. And so, so fish violate that in the way you just said, which is superposition doesn't work exactly right. They do change their behavior based on how predictable the stimulus is. But there's another part where they break linearity, which is if we have a slowly moving refuge and they're in the dark in particular, they do it in the light a little bit, but they also produce these back and forth ancillary movements, these what we call, these active sensing movements. So they produce a second set of frequencies that are not related to the input stimulus, the movement, the frequency of the refuge. So that's a major non-linearity. So
it's like a jitter on top.
It's a jitter on top of it. It's sort of like if you have smooth pursuit, or maybe you've got some sort of saccades on top of the tracking of the movement of some object, of some visual object.
And is it understood what kind of information that adds? It's like some sort of equivalent of a parallax?
So
jittering to measure
so
distances or something?
Well, that's been the focus of a lot of our work, and the experiments that I showed today really focus on that issue of why are they producing or these these nonlinearity, nonlinear movements, and how are they controlling it, and why are they doing it? So our hypothesis is that these active sensing movements are used to get more information about the environment that they're in. And so by investing energy and producing these movements, they are self-stimulating their sensory system in a way has more information than if they just relied on the linear interaction that they had with their environment. But that's just a hypothesis. Another idea could be that as they are less, maybe as low frequency movements are happening, they've got an internal noise generator that's always running, and so you see it more clearly at those frequencies. Or, there's a bunch of other things they could be doing. It could be a social signal for all we know. So in our early experiments, we tested the idea simply by changing the conductivity of the water. If you make the water more or less conductive, their electric field is more or less effective. And so as you make their signal less effective, they produce more of these back and forth movements. So that's a suggestion that they're using it to get information to acquire information about their environment. Another hint that it's being used to acquire information is that during the day, when their visual system is active, they produce less of these energetically costly movements, and when you turn the lights off, they generate them a lot more. So that's another suggestion.
Yeah.
And then you can do other things like change the structure of the refuge you can have a more featureless refuge, or one that has more features for them to detect, and see how much of these active sensing movements that they produce, to sort of guess that they're trying to acquire information using active sensing. The experiments that I showed today involved wrapping a closed loop control system around the animal, right? So the idea there is the fish is using its own movement to gain information, is our hypothesis. And so how do you test that hypothesis? We can't stop the fish from moving, right? And so what we did was to try and modulate what the impact of those movements on the feedback that it receives. If our hypothesis is the movement is to generate some feature of feedback, we can then alter the feedback that they get from the movement and see how they deal with that problem and test some hypotheses. So what we did was Noah's lab built a system that in real time uses a video feed from underneath that can detect the position of the fish, and it can simultaneously detect the position of the refuge. And so we can see the movement of the fish, and then you can run that through a computer control system that modifies the movement of the refuge. So if the fish moves forward, ordinarily, if the refuge is just sitting there, they're getting feedback, and exactly the same amount that they move forward is the same amount of feedback they're getting as they pass through this stationary shuttle. But then using this control system, we can move the shuttle. We could move it in the same direction as the fish at the same time, and we can basically eliminate the feedback. Or we could move it in the opposite direction to increase the feedback. And so if the goal of the fish is to generate feedback through its movements, maybe it'll adjust its movements based on how much feedback it's getting. And so the experiment worked pretty beautifully, actually, where if you make it so that the refuge moves half the distance in the same direction as the fish would move twice as far, but if you look at the amount of slip between the refuge and the fish, it was roughly the same as if the fish was in its normal condition. And then we could make the refuge move in the opposite direction, increasing the amount of sensory slip the fish got for any unit of movement. So now the fish would move less and maintain the same amount of sensory slip. So that seems like a really good demonstration that the fish are using these movements and they're modulating their movements based on the sensory feedback that they get. And then there's further details that we performed on the analysis, which is like what kinds of movements are they producing? It could be that the fish were trying to generate transients where they reverse direction. So maybe they get all their information from reversals, like going from forwards to backwards, backwards to forwards, and so they'll do that a lot. Another possibility is that they are trying to have a particular velocity of movement relative to the refuge. So they would have less of the transients, but more time spent at a certain velocity. And so our analysis seems to show that they have a preferred set of velocities and they generate long transients rather than having reversals. So that's great, and for us, and for me in particular, because I want to translate this into a neurophysiological experiment. And if I want to know what I'm looking for in the brain, I want to look for neurons that are going to respond or be tuned to the statistics of these movements that they're generating. And so this suggested that I shouldn't be looking for neurons that are detecting reversals, but I should be looking for neurons that have a range of selectivity for particular velocities. And so when we started doing the neurophysiological experiments in the freely behaving animals, we started looking for those parameters that were emergent during these behavioral experiments.
Wow. So the animal is making these movements as a form of exploration. It's exploring its electrical environment. And somehow these are the right movements to
generate. Well, I mean, there's lots of great examples of this. Like, when you're looking for, if you're going to the bathroom in the middle of the night and you've decided that you're going to do that horrible thing of turning on the light, and you can't see it, you're going to move your hand, and typically you move your hand in swathing kinds of movements, or sometimes people will tap, but I think most people are going to do sort of swatting kinds of movements until they hit the object. Your visual system, there's beautiful classic experiments where you tell people to look for different features in an image, and so they'll have visual saccades scanning the image, and they differ depending on, well, I want to know how old the people are, so you'll see all the saccades moving around their faces, or tell me how big the room is, and you'll see the movements of the eyes going differently. So you change your movements based on the information you're trying to get. Another classic example is, in somatosensory systems, where if I ask you the weight of something, you'll move your hand in one way, but if I ask you the texture, you'll move your hand in the other way, or the temperature, you'll literally hold it or squeeze the object in the other way. And so these kinds of movements are exactly analogous to that. They're moving their bodies to get the information they need to achieve that particular task.
So possibly by studying their movements, you can figure out what information they're getting, and that tells you what they want to know about the world.
Yeah.
Because right from the beginning, we don't just immediately know what it is they want to know.
Well, the thing that I like about this active sensing behavior is that we often imagine that the brain is there to control behavior. Like, I have a brain so that I can say these words. But active sensing sort of turns that on its head and says that I'm going to use my behavior to serve my nervous system or to serve my sensory system. So, these hand movements, each of the hand movements are directed to optimally, not optimally, but better stimulate certain receptors that are detecting that kinds of information. So you literally change your behavior to match the parameters of the receptors that are on your hand. And in the case of the refuge tracking, it's not the receptors that they're tuning the behavior to, but the computations that occur in the central nervous system. So I think these active sensing behaviors reflect the behavior being tuned to the parameters of the nervous system that exist.
Well, that's incredible.
Yeah, that's kind of
great.
a fun way of doing things.
Well, this is a good place to stop. We could keep going forever because this is so interesting, I think. But thank you for joining us, Eric Fortune.
Thank you so much.
And
It's a pleasure.
Francesco.
Thank you.
Todd.
It was fun.
And this has been Neuroscientists Talk Shop.